Unknown genetic sequences that don't match any creature on Earth. DNA that scientists can't trace to any known species. Code hidden inside human genomes that came from something we've never discovered. For over a decade, researchers have been finding fragments of mystery DNA inside modern humans, and they still can't explain where it came from. In 2010, a single finger bone discovered in a Siberian cave changed everything we thought we knew about human evolution. That bone contained DNA from a species no one had ever seen before. Scientists named them Denisovans, and here's the thing. We still don't have a complete skeleton. We don't have a skull. We barely have any physical evidence this species even existed. But their DNA is inside millions of people living today, especially across Asia and the Pacific. However, Denisovan DNA is just the beginning. Recent studies have found genetic sequences in Asian populations that don't match Denisovans, Neanderthals, or any other known hominin species. There's something else in there, something we haven't identified yet, and it's raising questions that challenge everything we understand about where we came from. If you're watching this because you want to understand the cutting edge of human ancestry research, hit subscribe right now. I cover the latest discoveries in genetics and human evolution every single week, breaking down the science that's rewriting textbooks. Now let's get into what scientists are actually finding. The story starts in 2008, inside Denisova Cave in the Altai Mountains of Siberia. Russian archaeologists had been excavating the site for years, finding stone tools and animal bones dating back tens of thousands of years. Then they found a tiny finger bone. It looked human. It was small enough to belong to a child. They sent it to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where a team led by geneticist Svante Pääbo extracted DNA from the bone. What they found didn't make sense. The DNA was human, but it wasn't modern human. It wasn't Neanderthal either, even though Neanderthal remains had been found in the same cave. This was something completely different, a third branch of the human family tree that no one knew existed. They called it the Denisovan lineage, named after the cave where the bone was discovered. Here's what made this discovery so strange. We've built entire museums around Neanderthal skeletons. We have hundreds of Neanderthal fossils from across Europe and Asia. We know what they looked like. We can reconstruct their faces. But for Denisovans, we have a finger bone, a few teeth, and a piece of skull. That's it. Despite having almost no physical evidence, their genetic legacy is massive. Studies published between 2010 and 2016 revealed that modern humans interbred with Denisovans, just like they did with Neanderthals. But while Neanderthal DNA is spread relatively evenly across non-African populations at around 2%, Denisovan DNA shows a very different pattern. It's concentrated in specific regions. Indigenous Australians carry up to 5% Denisovan DNA. Papuans in Papua New Guinea have similar levels. Populations in the Philippines, Indonesia, and other parts of Southeast Asia show significant Denisovan ancestry. But here's where it gets complicated. In 2018, researchers analyzing genomes from East Asian populations found something unexpected. There were stretches of DNA that looked like they came from an archaic human species, but they didn't match the Denisovan sequences already identified. They didn't match Neanderthals either. The genetic signatures suggested these sequences came from a different, unknown hominin group that interbred with the ancestors of modern Asians. This wasn't a one-time finding. Multiple studies using different populations and different methods kept arriving at the same conclusion. There's ghost DNA in modern humans, DNA from a species we've never found. And it's not evenly distributed. It shows up most strongly in people whose ancestors lived in Asia and Oceania. Sharon Browning, a statistical geneticist at the University of Washington, led one of the key studies that identified this mystery DNA. Her team analyzed over 5,600 whole genome sequences from individuals across Asia and Oceania. They used statistical models to identify segments of DNA that appeared archaic, meaning they diverged from the modern human lineage hundreds of thousands of years ago. What they found was evidence of at least 2 separate interbreeding events with unknown hominin species. One of these events involved a group that split from Denisovans over 300,000 years ago. This mystery population contributed DNA to people living in what is now Indonesia and Papua New Guinea. The other event involved a different unknown group that interbred with the ancestors of East Asians. Neither of these populations matches any known fossil record. We have no bones, no teeth, no skulls, just their genetic fingerprints left behind in living people. The implications are staggering. For most of the 20th century, the story of human evolution was relatively simple. Modern humans evolved in Africa around 300,000 years ago, then migrated out of Africa between 60,000 and 80,000 years ago, replacing all other hominin species they encountered. Neanderthals died out. Other archaic humans went extinct. Homo sapiens was the sole survivor. That narrative collapsed as soon as genetic technology advanced enough to sequence ancient DNA. We now know that modern humans didn't just replace archaic populations, they interbred with them. Neanderthal genes gave some modern humans stronger immune systems, adaptations to cold climates, and other survival advantages. Denisovan genes helped Tibetans adapt to high altitudes by improving their ability to process oxygen in thin air. And now we're discovering that the mixing was even more extensive than we thought. There were multiple unknown human species wandering across Asia, and our ancestors encountered them, interbred with them, and carried their DNA forward into the present day. But this raises a huge question. If these mystery populations contributed significant amounts of DNA to modern humans, why haven't we found their fossils? We've excavated thousands of sites across Asia. We've found Homo erectus fossils over 1 million years old. We've found stone tools dating back hundreds of thousands of years. But we haven't found skeletal remains that match these ghost lineages. One possibility is that we have found them, but we didn't recognize them. Fossil classification is hard. You need relatively complete skeletons to assign a species designation with confidence. Most ancient human fossils are fragmentary, a piece of a skull, a jawbone, a handful of teeth. Without DNA, it's incredibly difficult to determine if a fossil represents a distinct species or just an unusual individual from a known group. The Denisovan discovery proves this point. Before 2010, we had Denisovan fossils and didn't know it. That finger bone sat in a box for years before anyone extracted DNA from it. There could be dozens of other fossils in museum collections right now that belong to unknown hominin species. We just haven't tested them yet. Another possibility is that these mystery populations were small and lived in environments where fossilization is rare. Tropical rainforests, for example, are terrible for preserving bones. The acidic soil, high humidity, and abundance of scavengers mean that remains decompose rapidly. If an unknown hominin species lived primarily in Southeast Asian rainforests, we might never find their bones, even if they existed in significant numbers. Then there's the third possibility, and this one is the most unsettling. Maybe we haven't found them because they looked too much like us. If a mystery hominin species was physically similar to Homo sapiens, but genetically distinct, we might have fossil evidence and not realize it. We might have classified their remains as early modern humans or unusual Homo erectus specimens, never suspecting they were something else entirely. Recent discoveries suggest this might actually be the case. In 2019, researchers working in the Philippines discovered a new hominin species called Homo luzonensis. The fossils were found in Calao Cave on the island of Luzon and included teeth, hand bones, and foot bones dating back at least 50,000 years. The bones showed a strange mix of features. Some characteristics looked primitive, similar to much older hominin species like Homo habilis. Other features looked more modern. What made this discovery significant wasn't just that a new species was identified, it was where and when it existed. Luzon is an island. To get there, Homo luzonensis would have needed to cross open water, something that requires either boats or incredibly lucky accidents involving floating debris. And they were living there at the same time modern humans were spreading across Asia. This means our ancestors could have encountered them. So far, no DNA has been successfully extracted from the Homo luzonensis fossils. The tropical climate makes DNA preservation unlikely, but the discovery proves that small, isolated hominin populations were scattered across Southeast Asia and Oceania during the exact time period when modern humans were migrating through the region. If our ancestors encountered and interbred with these populations, their DNA would show up in modern genomes even if we never found their bones. And here's the thing that keeps geneticists up at night. The ghost DNA in modern humans doesn't just come from one or two mystery populations. Statistical analyses suggest there could have been several unknown hominin groups contributing genetic material at different times and in different regions. Each of these groups would have split from the main human lineage at different points. Some might have diverged 500,000 years ago. Others might have split off 700,000 years ago or even earlier. To put this in perspective, imagine trying to reconstruct a family tree using only DNA, with no photographs, no written records, and no names. You can see that certain people are related. You can estimate when their ancestors diverged, but you can't see their faces. You can't know their names. You can only detect their genetic shadows. That's the situation with ghost DNA. Researchers can identify genetic sequences that clearly came from archaic human populations. They can estimate when those populations split from the lineages that led to modern humans and Neanderthals and Denisovans. They can track which modern populations carry the highest levels of this mystery DNA, but they can't name the species. They can't describe what they looked like. They can only confirm that something was there. Emilia Huerta Sanchez, a population geneticist at Brown University, has spent years studying the Denisovan gene variant that helps Tibetans survive at high altitudes. This gene, called EPAS1, regulates red blood cell production and allows Tibetans to thrive in environments where most people would suffer from altitude sickness. The gene is incredibly rare in other populations, but it shows up in over 80% of Tibetans. When Huerta Sanchez and her team traced the origins of this gene, they found it came from Denisovans. But here's the fascinating part. The Denisovan finger bone was found in Siberia, thousands of miles from Tibet. Denisovans clearly had a much wider geographic range than the single cave site would suggest. And if they ranged across Asia, they would have encountered many different environments and potentially many different hominin groups. This brings us to one of the most important discoveries in recent years. In 2019, a team of researchers led by Diendo Masalani analyzed DNA from a 90,000-year-old bone fragment found in Denisova Cave. The fragment came from a teenage girl whose mother was Neanderthal and whose father was Denisovan. This was direct proof that different hominin species interbred and produced viable offspring. But the analysis went further. The girl's Denisovan father had Neanderthal ancestry in his own genome. That means his ancestors had also interbred with Neanderthals, but in a different time and place. The mixing between human lineages wasn't a rare accident. It was happening repeatedly across tens of thousands of years. And if Neanderthals and Denisovans were interbreeding, and modern humans were interbreeding with both groups, then it's almost certain that other unknown hominin populations were part of this genetic exchange as well. So what does this mean for people living today, especially those with Asian ancestry? For one thing, it means your genome is more complex than you probably realize. If you have East Asian, Southeast Asian, or Pacific Islander heritage, you likely carry DNA from at least 3 or 4 different hominin lineages. Some of that DNA comes from Neanderthals, some comes from Denisovans, and some comes from mystery populations we've never identified. This ghost DNA isn't junk, it's functional. It codes for proteins. It affects how your immune system works, how your body responds to viruses, and even how your brain develops. Studies have found that some Denisovan gene variants are associated with improved immune response to certain pathogens. Other variants affect metabolism and energy production. In 2020, researchers discovered that a gene variant inherited from Neanderthals increases the risk of severe illness from COVID-19. The variant is most common in South Asian populations, where it shows up in around 30% of people in Bangladesh and 50% of people in some parts of India. This single genetic difference, inherited from a species that went extinct 40,000 years ago, is still affecting human health today. The same is true for ghost DNA from unknown species. Some of these genetic sequences have been positively selected, meaning they provided a survival advantage and became more common over time. Others might be neutral, neither helpful nor harmful. A few might carry increased disease risk, but we won't know the full picture until we identify exactly where this DNA came from. That's the challenge facing geneticists right now. They're trying to build a complete picture of human ancestry using incomplete information. Every new genome they sequence adds more data. Every new statistical model gets them closer to identifying how many mystery populations contributed DNA and when those interbreeding events happened. But without fossils, without physical remains, it's like assembling a jigsaw puzzle in the dark. Let me give you a specific example of how this detective work actually happens. When researchers analyze a genome looking for archaic DNA, they're searching for segments that show unusual patterns. Modern human DNA from people living today has certain characteristics. The sequences have evolved over the past 60,000 years or so, since our ancestors left Africa. They've accumulated specific mutations. They show patterns of recombination where chromosomes from mother and father get shuffled together during reproduction. But archaic DNA looks different. It diverged from the modern human lineage hundreds of thousands of years ago, which means it accumulated different mutations. When you find a stretch of DNA in someone's genome that has a completely different mutational signature, that's a red flag. It suggests that segment came from an archaic population. The problem is determining which archaic population. Scientists have reference sequences for Neanderthals and Denisovans now. They can compare unknown DNA segments to those references and see if there's a match. But when a segment doesn't match either group, that's when things get interesting. That's when they've potentially found ghost DNA. Here's how they narrow it down. First, they look at how divergent the sequence is. The more mutations that have accumulated, the longer ago that lineage split from modern humans. By counting mutations and using what's called a molecular clock, researchers can estimate when the split happened. Next, they look at the geographic distribution. If the mystery DNA shows up primarily in people from Indonesia, but not in people from Europe or Africa, that tells you something about where the interbreeding happened. Then they look at the frequency. If 5% of a population carries a particular archaic gene variant, that suggests the interbreeding happened multiple times or that the gene provided some advantage. If the frequency is very low, it might have been a rare event, or the gene might carry a disadvantage that's being weeded out over time. Finally, they compare the mystery DNA to known hominin fossils. Even without extracting DNA from old bones, you can sometimes make educated guesses based on where and when those fossils existed. If you find ghost DNA in modern Filipinos, and you know that Homo luzonensis existed in the Philippines 50,000 years ago, it's reasonable to hypothesize there might be a connection. But it's still just a hypothesis until you can test it directly. All of this requires enormous computing power and sophisticated statistical methods. you're dealing with billions of genetic base pairs across thousands of individuals. You're trying to detect signals that might represent less than 1% of someone's total genome, and you're doing it without knowing in advance exactly what you're looking for. Despite these challenges, the discoveries keep coming. In 2023, a team analyzing genomes from the Andaman Islands, a chain of islands in the Bay of Bengal between India and Myanmar, found evidence of archaic DNA that didn't match any known hominin group. The Andamanese people have been isolated for tens of thousands of years, which makes their genomes particularly valuable for studying ancient ancestry. What makes the Andamanese findings significant is that this population split from other Asian groups very early, possibly over 40,000 years ago. If they carry ghost DNA, it means the interbreeding with unknown hominins happened before that split. That pushes the timeline back and suggests these mystery populations had a wider geographic range than previously thought. Think about what the world looked like 50,000 years ago. Modern humans were just beginning to spread out of Africa and across Asia. The climate was different. Sea levels were lower because so much water was locked up in ice sheets. What is now the islands of Southeast Asia was mostly connected by land bridges. You could walk from mainland Asia to Java, Borneo, and other islands that are now separated by ocean. In that world, there could have been dozens of isolated hominin populations. Some might have been descendants of Homo erectus, who had reached Asia over 1 million years earlier. Others might have been entirely different lineages we don't have names for yet. And as modern humans moved east, following coastlines and rivers, hunting and gathering, they would have encountered these populations. Sometimes the meetings might have been hostile, competition for resources, territorial conflicts, but other times they interbred. The genetic evidence proves it happened multiple times with multiple groups. And here's something that doesn't get talked about enough. When we say that modern humans carry Neanderthal or Denisovan or ghost DNA, we're talking about successful interbreeding. The offspring survived to adulthood, had children of their own, and those children continued the line for thousands of generations. That's not guaranteed with hybridization. Many hybrids are weak, sick, or sterile. The fact that archaic human DNA is still present in billions of people today means those hybrid individuals were healthy and fertile. They weren't evolutionary dead ends. They were integrated into human populations and became part of the story. This has implications for how we think about human behavior and culture. If your ancestors were successfully interbreeding with other hominin species, they must have seen those individuals as potential mates. That suggests a level of social integration that goes beyond hostile encounters. Maybe there was trade. Maybe there were alliances. Maybe families from different groups lived together. We know from archaeological evidence that modern humans weren't the only ones making sophisticated tools or creating art. Neanderthals were making jewelry and symbolic objects 100,000 years ago. There's evidence they cared for their sick and elderly. They buried their dead. These were people, not mindless animals. And if Neanderthals had that level of cognitive sophistication, it's reasonable to assume Denisovans and other archaic humans did too. So when modern humans encountered them, they weren't meeting alien creatures. They were meeting people who looked different, maybe spoke different languages, but who were fundamentally similar in their capacity for thought, emotion, and culture. That changes the way we should think about those encounters. Now let's talk about what this means for the future of the research. DNA technology is advancing incredibly fast. 10 years ago, sequencing a complete human genome cost around $10,000 and took weeks. Today, it costs less than $1,000 and can be done in a couple of days. That means researchers can analyze larger and more diverse populations than ever before. There are ongoing projects right now to sequence hundreds of thousands of genomes from people all over the world. The goal is to build a comprehensive map of human genetic diversity. And embedded in that data will be more ghost DNA, more mystery lineages, more evidence of encounters we don't have names for yet. At the same time, scientists are getting better at extracting and analyzing ancient DNA from degraded fossils. The techniques improve every year,